Okay, well, sorry about that. So let's just begin. I want to start out with a little kind of intro thing, and then we'll go into the presentation, which really has two parts. Given the timing thing, maybe we're never going to get to part two. But I'm now going to show something. I'm going to press a button, and you're going to see something on the screen very quickly, and then I'm going to ask you, what did you see? Okay, so take a look. Everybody looking? Here we go. So now the question is, how many people don't know what the heck they saw? If you don't mind raising your hands here. Some heart card, some diamond, 5 of hearts. Everybody, 5 of diamonds, maybe. Okay, so now I'm going to do the same thing. Look again. What did you see? Okay, so now, don't know. Some spade card, and some club card. Still no answers. The 4 of spades. A lot of people. 4 of clubs. Nobody. So the deal is, what you saw was this card, which is a 4 of hearts, except it's black instead of red, right? And this is actually, I'm doing something here that was done by a pretty famous developmental psychologist named Jerome Greener back in the late 40s, where they used a tachistoscope, which is something that projects things for different amounts of time on the screen, and then you look and try to identify what you saw. And so they were able to control the length when we weren't here. But basically what they found was, they used, of course, regular playing cards, but they also threw in these so-called anomalous cards where they, weren't a card that you expected. And they found that to reliably identify an anomalous card, it needed to be exposed on the screen for 4 times as long as a normal card. Okay, what's the take home? What do you think this says something very profound to me. What does it say to you? Anybody? Thoughts? Yeah.
Unexpected things need more processing time.
Yeah, I think unexpected things need more processing, or mainly if you're expecting something, the flip side of what you say, if you're expecting something, it almost governs what you can see. That it takes so much longer to realize, oh, that's not what I was expecting. And so basically we would call this bias, right? And of course we're talking about ethics, and this is one of the issues that we'll be talking about is bias. So today's topics, I'm going to talk a little bit about 2 challenges that are going on, and then second is the most important ethical issue of our time. So we can contemplate what that might be. So what are these 2 challenges? The first one is what was summarized here and something that people have been hearing, which is NIH funding kind of dictates what goes on in biomedical research, and it's kind of boom and bust. Like it goes up, George W. Bush had a program to double the NIH budget over a 5-year period, a tremendous expansion, a number of people doing research expanded, and then it got cut back. And all of a sudden there's crunch time for trying to get the money, and if you talk to PIs at all these days, you find out that they're feeling like they write grants all the time because it's very hard to get them. And so this boom and bust cycle makes for a lot of anxiety, a lot of tension, a lot of pressure. And these guys, all of whom are super big heavy hitters in biomedical research, wrote this very influential article in, by now, maybe over a year ago. An unsustainable, they described the U.S. they start out saying what you probably all know, which is that the U.S. biomedical research community is the best in the world, has done remarkable things, and led to tremendous advances that have been applied to medicine. But things are kind of going a little bit haywire with all of this pressure, and they said that it's creating an unsustainable, hyper-competitive system that's discouraging even the most outstanding prospective students from entering the profession, and making it difficult for seasoned investigators to produce their best work. So one of the challenges, so this causes pressure, we're going to look at that, but it also is a challenge to restructure things in some way to alleviate this, to make for people to have meaningful, productive careers. Okay, so what's the second challenge? And this one I think is something that's less widely appreciated, although becoming much more well-known, and a serious problem that could be a serious problem for all of you if you're interested in going into research. Namely, a high proportion of published papers are unrepeatable. And I'll show you a little bit of the data for this. So, not reproducible, wasting researchers' time if you're trying to follow up on what somebody did or reproduce it, and resources, and threatening to undermine congressional support. If I'm in Congress, my job one always is trying to, get the things that I want funded, and cut other things to make it possible. Money is always tight. If I start getting the idea that research is not repeatable, that like 60, 70, 80% of what's published is essentially not good or not that, not reproducible, I'm going to start thinking, well, let's cut NIH's budget, right? So this affects all of us. And it affects us in a more profound way than simply getting money, which is waste people's time when we could be doing productive things to really contribute to the health mission. Okay. So, not a good thing. So here they said there's growing evidence. Here's some of the growing evidence. This was a very influential paper published back in 2005 by this guy, Emeritus. Why most published research findings are false. Most published findings are false. And he went through arguments and some data about this. Okay, that was one kind of thing. A lot of people are familiar with that. You get national press seeing it. This was actually lead editorial in the New York Times from June of last year. What happens when scientists cheat? So fraudulent things. Here's another one. So this is a really significant one. It became kind of publicly known, and this alludes to this here. This says Bayer, people make Bayer aspirin and lots of medical things, halts nearly 2/3 of its target validation projects because in-house experimental findings fail to match up with published literature claims. So like pharmaceutical companies want to develop drugs that they can market. If there's a paper published that says, oh, we've discovered that this protein is expressed in a certain kind of cancer, and there's a certain compound that inhibits its action, and this could be a good target for drugs. So then the drug company will try to repeat that because they can perhaps develop derivatives of that might be better or at least are patentable. And they find that it says here, nearly 2/3 of those trials fail to repeat the published results, even when they're performing precisely the methods described in the paper. And I've actually heard more recent estimates of this that make the number higher, 80%. A buddy of mine who graduated from the College of Medicine and is a big shot with this company that does eye drops and contacts and things, said that in their case he finds it's like 90% of the things that cannot repeat. Okay, so this is disaster. Okay, and so what do you think is the most important factor? Pressure causes more cheating, it's bias, it's bad experimental design or statistics. So show of hands, how many people think that the most important of these things is likely to be pressure on investigators? Yeah, a lot of you. How many people think it could be bias? I think maybe you can, the nice thing about this, unlike the clickers, is you could vote more than once if you wanted to. What about bad experimental design or statistics? A few. Okay. So let's look at some of these things, right? There, 0, 0, 0. Okay, cheating. Let's see. Most papers, what do you think? Is this true or false? Most papers are retracted, that is, retracted like, not true, we take it back, from low impact or unrefereed journals, most low quality. So in other words, if there's like bad science out there or cheating or something, is it getting through, who's publishing it? Is it low quality journals? Most of it's coming from these things that are unrefereed or low impact, or is it high quality? How many people think it's true that it's really mostly going to be in the low quality journals? And how many people think in the high quality journals? Good for you, because that's true, even though it's really surprising. This thing shows you along the left side there the impact factor of a journal, how important it is, how many times the papers are cited. And you break it so the things that are high up there, like Lancet, Nature, Cell, and Science, are the premier journals. This axis down here is a retraction index where they take number of retracted papers divided by total number of papers published and so forth. And basically what you find is these journals here, Nature, Cell, and Science, the most high impact journals, have the highest proportion of retractions. Over here, these are very good journals, not quite as high impact as those, a lower form. And over here is where I publish. So why? What could be the reasons for this, do you think? All you future researchers. Hypothesis generators. Yeah, back here.
Well, maybe like some of the principal investigators who are expected to be publishing in these like high impact journals are feeling that pressure that we've been discussing before, and feel like if they're not producing like quality journals, and they're not going to by nature get published, and so they feel like they have to forge this, or forge or like kind of like cheat.
Right. So it could be fraudulent, or it could be cutting corners or something, but basically it's the pressure, as we were saying, that's acting on them. Is there another possible reason, you think? Yeah.
It could be that these publications are trying to get more readers and trying to publish, I guess, like more stimulating, more interesting pieces of work.
Yeah, so this is interesting. People are trying to publish good stuff and they're kind of juicing it up a little bit, and you're saying, and the journal is trying to get good stuff, and they're like getting the sexiest stuff, which may not be as proven. Very nice. How about any other reason? There's another possible reason, I think. Kind of more methodological. Yeah.
The higher end journals are, since they have the higher reputation, they're more likely to want to retract something once it's become.
This is slightly different than what I'm thinking of, but it could also definitely be the case. They may be more careful and retract things when it's not totally proven or something to be careful. Yeah.
I was just thinking if I was going to reproduce a procedure and experiment, I would not want to reproduce one of the lower quality journal, and I would look to a higher quality journal, and thus they just find more things that are wrong.
I think what you're saying is very much like what I'm thinking and what they proposed in the paper where they analyzed this, which is that the higher quality, these are the high impact, these are the ones that people read and scrutinize, and so more people look at it and more people will try to repeat it or will say, wait, what about this little thing here? That there's just more attention paid, and so more mistakes are caught, even though there may be mistakes in the others, which is kind of maybe more like in who cares territory. So, good. All right, so it's a pretty interesting issue, but kind of surprising. And of course, these are the things that make big headlines and that undermine the public's confidence in science, which is not great. A good thing. So we talked about that. Now, I said cheating is a problem. You were saying maybe they were kind of falsifying, or maybe it's just sloppy or whatever. This is a meta-analysis where they took a bunch of different kinds of surveys that people had done trying to find out how much do people make up stuff, what kind of things do they do wrong. Polling scientists, right? And then pulled it all together to try to get some kind of consensus view, because the questions are asked in different ways and so on. And basically, here's the thing. A pooled weighted average of about 2% of scientists admitted to having fabricated, falsified, or modified data or results at least once. Serious form of misconduct. This is a terrible thing. I mean, 2%, that's not good. And it could be higher. Maybe this is what people are admitting, although it's anonymous and so on. So it could be a little higher. But if we're saying that 70 or 80% of the papers, can't be reproduced, this is a bad thing, but not accounting for everything. Okay. Pressure. Everyone else does it. I won't be competitive if I don't do it. Right? What fields do we know that this applies to? Maybe it applies to publishing research. What else does it apply to where people feel like, well, that's wrong, but everybody else is doing it, I need to do it too?
Like athletics, like sports.
Yeah. I think probably this happens all over the place, but certainly that would be the more obvious one, right? So the use of steroids in baseball, and blood doping in cycling, and probably lots of other kinds of things. And I think it's easy to be high-handed and moral and so forth, and denounce people who cave into this pressure. But I think realistically speaking, when you talk to football players who say, I have a family to support, and if I'm not playing on a level playing field with other people, I'm letting down my family, and so forth. And, so it's not. I don't think this is an easy issue. And in fact, there was an interesting article here in Sports Illustrated, where 10 years after, so there was this guy Ken Caminiti, who was one of the first people to expose the use of steroids in baseball. And 10 years later, they kind of took a retrospective look. And in this particular article, what they did was he examined the playing careers of 4 right-handed pitchers who had tried out for the Minnesota Twins organization in the mid-90s. And none were drafted higher than the 4th round. One of the 4, however, took steroids, and he was the only one who ever reached the major leagues. And it says, he was 6'6" and 180 pounds as a senior, had a fastball that he threw at around 85 miles per hour and was drafted in the 14th round. After using steroids and other performance-enhancing drugs, he began throwing his fastball up to 95 miles per hour and at one point weighed 248 pounds. He got traded to the Yankees, played in the World Series where they won the championship. The other people, when this guy interviewed them, said, I don't know how I didn't realize what was going on at the time. That this guy had cheated us. That, he's all of a sudden starts throwing so much harder. All of a sudden puts on all this weight. But I didn't really know, they said. But they're angry, of course, because they feel like they played fair and didn't win the big prize. This guy went to the major leagues, won a championship. I don't think this is always the case, but this guy actually suffered very serious remorse and at one point tried to commit suicide. But I think that we know other figures who are not so remorseful. And I think that this highlights a very challenging problem in circumstances where pressure is put on you to do what you believe at least that everyone else is doing, and what in fact everyone else may be doing. And it's something that you basically can call on your personal resources. It's not like there's a pat answer for how to avoid this. This can be a big problem. But I'm pointing it out. Okay. So that was cheating. Let's look at bias a second. So we looked at those cards before. So now let's do it again. What do you see here? All right. Okay. A who knows what card? A spade card, a heart card, the 7 of spades, 7 of hearts. I don't know how you're interpreting the answers here because, of course, what you saw was a 7 of spades, only it was red, right? So in a sense, the correct answer was the first one, which was like, I don't know, because it wasn't any of the real choices. But I think you've got the picture, and I think most of you, this time, were able to recognize that this was a funky card, and were able to see what it was in the same kind of time frame that originally, when you did it, you weren't able to. And because you now had you were open to the possibility that this could be a correct answer, whereas originally it didn't seem like it could be. And I mean, experiments are like this. You can go into it, or you may have a strong idea, or your PI may be telling you that there's a theory, an hypothesis in the lab that they've been supporting and their grants depend on and so forth, in which the expectation is that you're going to get a certain result. And if you don't get that result, I can assure you that first year and second year graduate students, and maybe more, when they don't get the result that the PIs expect, the PIs think those guys haven't learned how to do experiments yet. They don't think, oh, my theory's wrong. They think, you're not doing it right. Try this, try that, try the other. Well, that doesn't hurt to try those things because it could be you're doing the experiments wrong. But ultimately, it often, or sometimes at least, turns out, of course, that you were getting the right answer. The theory was wrong, at least in this respect. But there's such a strong pressure and presumption that it's going to come out the way people think that it takes a lot of confidence and a lot of determination to overcome that. And typically you don't have those when you're first starting out. It takes a little while to develop that, but it's an issue. Okay. All right, and this is basically, right, this is the answer. I like this expression, right? I'll see it when I believe it. Normally, of course, you say I'll believe it when I see it. So I'll see it when I believe it, it kind of describes our experience with the cards. You didn't see it originally, and then when you realized or believed that that could happen, then you could see it. So, an interesting thing. Okay, so if you have this kind of bias, how do you control for it? And we won't talk about these things, but these are significant deals. Statistics, when applied correctly, can really help you eliminate, determine whether the experiments are really coming out that way or it's your bias in it. Falsifiability, designing experiments that are not just proving your hypothesis, but have the chance of disproving it. A key thing here are blind tests and double-blind tests, which I'm sure you're kind of all familiar with this idea, that you don't know what the sample is, so you can't exercise any bias. And even the person who's giving you the samples doesn't know what sample is what, so there's no way of telegraphing what they are. And so these things, when adhered to rigorously, can really avoid a great deal of the kinds of troubles you can get into if you kind of have a hypothesis and you start looking at your data as if it fits it rather than as if in a challenging way. And so I say here that these methods are routinely used by the best researchers, but are disdained by some vain fools, which are, kind of harsh words. But I mean, I have heard people say, "Oh, I don't need to do double-blind things. I know how to score these. I can tell what's what." And I'm just here to tell you that I consider these people to be vain fools. It's so easy to fool yourself when you want a certain result. It's not just that it's so easy; it's hard not to. And you have to take very serious precautions not to, and you should be kind of the square person who does the things the right way. If the results don't come out when they're properly double-blinded and the statistics are being analyzed and so forth, those aren't the results, right? I mean, then the results are not valid. So, very important. Okay. So why most published research findings are false, and this guy's paper, where I said, he kind of analyzed the reasons, and he said that there's a combination of things. Poor statistics were very important, he thought, that bias and methodological errors, and probably cutting corners applying to some of it as well. And I think that responsibility, then, when you kind of put this all together, what I think the ethical take-home on this has to do with essentially responsibility, and where I think that it means you have to maintain your personal integrity to avoid pressure, to stick by what you think is right or true. And that's hard, can be in many circumstances, can be really hard. I think we all know that whistleblowers in this country more often than not get punished than rewarded for having rung the bell on somebody even when they're absolutely right. It's not easy. Raise your conceptual, experimental, and analytical skills to the highest level so your results can be trusted. So I think what we're saying here is, in addition to moral integrity, the only way you can do good experiments is if you really use the best possible techniques, the most rigorous analysis, and try to test things every way from sideways so that you know they're true, that you're really putting everything into it, that you're sharpening your skills all the time. I consider that to be almost, in this kind of endeavor, to be almost an ethical kind of responsibility. And then contribute to a research structure that promotes integrity, excellence, and fairness, and provides personal and professional rewards and satisfaction. We have now this high-pressure cooker kind of situation with the funding. That'll get fixed when the pendulum swings the other way. But it's like we should also be working to try to, make a structure that is fair, produces good results, and makes people satisfied with what they do. And part of your job as you move into the structure is to help mold it and make it that way. So there. How are we doing time-wise? Badly?
No, you're doing great. It's 5:30 right now.
Okay, so
Yeah.
the same guy, John Ioannidis, also published a paper. This was helpful, right? How to make more published research true. What can you do? And he said some of the things that I just said. And in addition, these things, he says, in addition to these things about how you do the work in the lab, optimal interventions need to understand and harness the motives of various stakeholders who operate. Like, what about the structure? What are people's needs? It's like, if you're under such pressure to publish papers to get promoted, then that's going to distort what you're doing. And maybe you can make changes in the system that will reward more accurate or careful publication than bulk publication and so forth. So he's talking about some systemic kinds of things that would address the environment as well. So I think that's important. And I'm going to skip this, just so we can move on. Okay, so now here comes... I said it was the most important ethical challenge in our time in the 21st century. So now this time we start saying, well, that's my view of what the most important is. Anybody, what do you think? Anybody have ideas here about what are the most important ethical challenges of our time? There's a bunch of them, right? I don't know what the most important is. What do you think are some of the big ones? Everybody's happy. No challenges. Everything's fair. Everyone's happy. Life is great. People are wonderful. Go on. Yeah.
Like the distribution of wealth.
Ethical distribution of wealth. Inequality, right? Some people are doing super, super well, and some people are pushing rubber rafts off the shore and trying to make it across the Mediterranean. Yeah.
Governmental surveillance.
Governmental surveillance, yeah. Individual privacy versus oversight and surveillance. That's a very important issue. It's kind of funny. I mean, one of the things I grew up with was nuclear power, right? The atomic bomb versus nuclear power is a huge issue and so on. Somehow, no terrible consequences yet, but that was the biggie in the last century. Any more notions? Okay. Well, do you think that what I'm about to talk about was one of the ones we just mentioned? No, right. But I think there is an issue within, when I said nuclear power, I mean the last century was really, the moral issues dealt with physics, with the discovery of atomic power and things, and the potential to extinguish mankind. And now I think that the challenges are really much more biomedical, have to do with reproductive rights, with, and the kinds of things that you can do to human beings. And so what I'm referring to here is germline gene editing. This is really an issue that in your lifetime, I think, will become increasingly significant and controversial and really discussed. And so it, I think, is useful for you to learn something about it mechanically, technically, as to what's going on, but additionally to think about the issues that are there, which I think are not simple. And so we'll just do a quick kind of brush through a little bit. So it's big in the news. There's like just tons of things everywhere all the time. And there have been just recently a couple of big conferences of largely scientists, but with some other kinds of public figures and ethicists discussing the idea of germline gene editing and mostly kind of favoring the idea of having a moratorium on it, not doing it until we know more. But so let's look at it. It's kind of funny that there's so much controversy now because changing the genome has been going on for a while. It's just that it used to be kind of clunky. So I said, here's kind of old style. Here's like you have a person, here's 2 sets of chromosomes, and on this chromosome you have a gene, you have 2 copies of this gene, and it's normal and it functions, and your life is good. But over here is a person who has inherited messed up, defunct copies of this gene. So for example, this could be a person with cystic fibrosis, which is a recessive gene. You don't have it, you're a carrier, but you don't have the gene if you only have 1 copy. But if you have 2 copies, you have no functioning protein, and you end up having, this terrible disease. And there are a lot of recessive lethal diseases like this. So the old style on gene therapy is you take a copy of the normal gene and put it into a little plasmid and then deliver it in a virus or by transfection or something into cells, and you'd end up with, you still have the same 2 dead copies, but you now have a good copy somewhere, and this thing makes the protein. If it went into a place where it could function and everything was okay, it could make the protein and maybe compensate so that the person would be a carrier instead of a victim of the disease. But it's very technical oriented. I mean, there were a lot of, this was going to be a big deal in the 90s, and then in about 1999 and into the 2000s, there were a couple of people who died in gene therapy clinical trials because of complications that were, technical things that were not anticipated, and it just poured ice water on the whole project for a while. But so it has been happening, but there's a new version now in which when people now talk about this as gene editing, and the idea is that you're correcting mutations or variations in chromosomes that cause disease, either in, now somatic cells, right, are like all the cells of your body except your germ cells. So this, doing gene therapy or doing gene editing in somatic cells is maybe a little controversial, but not so controversial. Most people, I think, are in favor of it if it can be made safe, but some are not. But the thing that's very controversial is making these changes in the germline so that you really change what's inherited from generation to generation. You're making permanent changes. The somatic cell repairs are just you personally and not passed on to anyone. And the reason it's all become so, has everybody heard of CRISPR? Is this how many people have heard of it before I said the word? Yeah, so few, that's good. You'll be starting to see this in headlines and hearing it on NPR and stuff once you kind of recognize it. So this system, which I'll describe very, very briefly, suddenly made this whole thing easy. This was really hard, this clunky old stuff. Easy, fast, and inexpensive. Literally, like, if I wanted to do this, I know enough molecular biology and could go into the lab and could make things happen doing this. It doesn't take like super experts anymore. So it's just that it's going to become super widespread and instantaneous, and people can do it underground and so forth. Yeah?
We're using CRISPR in my lab actually, like for my project. So.
Yeah, there you go. So you're doing it.
Yeah.
Cool. All right. Of course, humans is another deal, right? People are very chary about doing that. But, okay. So basically what's going on here is you have 2 key components that are all to this. There's a so-called guide RNA where you make, the way you have a certain sequence that you want to mess with. You know that sequence, and so now you make a little thing called a guide RNA, which corresponds to that sequence. That guide RNA hooks onto an enzyme that cuts DNA and takes it to the place. So here you have the big thing here is the nuclease that will cut the DNA, and this thing over here is the guide RNA that's matching up with this sequence in the DNA. So it's like, you have a sequence, you want to go, I'll show you. I have an example. Okay, so here's a gene, and I put a little asterisk there to say, oh, there's a mutation, and we want to fix it. So what happens is you want to snip it out, and so what you use is this guide RNA and this nuclease, and you put them into a plasmid, like in the other case, but you just put this plasmid in here, or you can inject them themselves, and the plasmid will make this guide RNA, and it'll drag the Cas9 over there, and it'll cut it. So, kaboom, you now have picked the exact spot you wanted, and it cuts the DNA in that spot. If you wanted to do a different spot, you just make a different guide RNA, and it drags the Cas9 over there and cuts it over there. Okay, well, that's just cutting it. You've cut it out here, but how do you fix it? And what goes on when this happens is that cells have repair systems of their own, and they have 2 big ways of doing this. One of them, so here's the illustration of the 2. So on the right here, this is what's called non-homologous end joining. This cut thing here, it kind of nibbles away a little bit and fills things in or whatever, and then just kind of like jumps the 2 ends together and glues them. And that ends up with something that's called disruptive, right? Typically, you'll make another kind of mutant when you do this. This doesn't help you much. What you want is an accurate repair of that. But the way you can do that is you put in the copy of what you want. So here, a repair template. So here's now something that's essentially exactly the same as this, except it's been repaired, it's got the right sequence that you want, and you get this process called homologous recombination, which occurs in cells, will cause your repair template to blip in there and repair the gene that was there, so you now have this edited gene that's fixed. So you just take these three components, you put them in the cell, you wait, and a little while later you can sequence it and say, aha, yes, nice. If you're getting the things done by this homologous recombination instead of this. Okay? So this is the basis of this thing. You can do this in somatic cells, you can do it in germline cells. How are we doing time-wise?
5:39.
Oh, perfect. Okay, so we'll finish this off very fast. I'm going to show you an example. So I say, okay, how about using gene editing to treat a specific human disease? Now, in fact, what I'm going to show you is work done in mice, of course. But, so Duchenne muscular dystrophy, as you may know, I'm going to leave this thing with you. If you want, you can click on this and you'll get a little thing, if you don't know about it. Terrible disease, affects mainly males. And when kids get to be about 3 years old, all of a sudden they have a little trouble walking, and 4 or 5, they can't lift up, and eventually they're in wheelchairs and they tend to die by mid-20s. Start out being normal, happy kids and end up dying, very young, and not so great. So something that people really would like to be able to fix, and it's caused by a mutation, and so it's susceptible to gene therapy kinds of repair. So you could do this, you could do the repair either in muscle, that would be somatic cell gene repair, or you could do it in germline. So these guys, actually, this Olsen lab, did it both ways in 2 publications. I'm just going to show you the germline because this is kind of the more controversial thing. So the gene, there was a mouse isolated some long time ago that had all the characteristics of Duchenne muscular dystrophy, and it was X-linked in the mouse, just like in humans. And eventually, when the gene was cloned, they found out that, in fact, it's the same gene that's altered. There's a specific mutation in the mouse, and essentially, it's a Duchenne muscular dystrophy mouse. So this is the system they used to try the gene therapy on. And if you look, the gene, the DMD gene, Duchenne muscular dystrophy, it actually codes for something called dystrophin. It's a huge gene, a whole bunch of exons. Here's 22, 23, 24. And then in this one exon, in more or less the middle of the gene, there's a single change in a nucleotide that converts this thing into a stop codon, and so you get a protein that's coming along here about halfway done and suddenly truncates. It's just half a protein doesn't do its job. The muscles degenerate. So what they did was to say, okay, now we're going to make a repair DNA sequence that will correct this back to the normal sequence. And they put in some other changes so that they could analyze whether they really put their gene, whether their correct gene, and these changes don't change the amino acid sequence, but you can use it to check. Okay. So the strategy was like this. They crossed a couple of these muscular dystrophy mice and got fertilized eggs, and then just added in here the Cas9, the enzyme that cuts the specific guide RNA that targets that spot where there's the mutation, and the specific sequence that will repair that mutation. So they inject these things into the egg, and then they reimplant these eggs into a superovulated female pseudo-pregnant mom, and they'll put 10, 15 or so of these into one mouse mom and get a bunch of pups born. It says here corrected, but they analyze these then to see, did it correct the gene or not? And as you recall, here's the little snips again, when they took this exon and did the repair, if they got homologous recombination, then you should get a completely repaired thing. If they got non-homologous end joining, it might be messed up. In fact, they got some good ones from this too, but not worth bothering. But what they found was they analyzed, they eventually got 7 pups born that had come out of this thing. 7 of them had been repaired by the homologous recombination and were exactly normal with the repair thing that they put in. So they were fixed. Another 4 of them were fixed by the other method, the non-homologous end joining, but it fixed in such a way that the protein still functioned, without going into detail. But one thing that happened was they got genetically mosaic mice. If the repair happened in that original cell, then as that cell began dividing and produced the entire embryo, all the cells would be repaired. But if that cell divided once or twice before the repair occurred, then only the cells here would be fixed and all these other cells would not be fixed. And so they'd have a mixture, a mosaic mouse. And they got, in the different mice, they found that some of them had, almost all the cells were fixed, some of them had very few cells were fixed, so they can kind of look at the difference. And you can see that difference here. Here is a cross-section that looks at muscle fiber, and these, the green staining around each of the fibers is the protein that you have to have, which is missing in the muscular dystrophy mouse. But in these guys that they had fixed, this was a partially, only some of the cells had it, but you can see you got some of these fibers there. But these guys, even less than half of them were fixed, but you got essentially complete fixing of the muscle. And over here where it was almost done, you got re-expression of the protein, the muscle fibers looked normal, and when they did grip strength, where they have the mouse grab onto something and they yank on the tail gently to see how much strength it takes to pull the mouse off of the little bar or something. The mice that had been, like these mice here, the gene-edited, germline gene-edited mice, were almost the same in terms of grip strength as the wild-type mice. So basically, in other words, this worked. They corrected the muscular dystrophy in the mice. Not fully, and it was different in several shots and corrected in different ways. So, yeah, it worked. It was variable. Sometimes it was corrected by one method, sometimes by the other. Wasn't fully effective because they had these differences in the mosaicism. So now, the question would have been, are we now ready for human germline editing, technically? How many people think, yes, we're ready, this worked in mice, we're ready to try it in humans? And how many people think, not yet? Yeah, not yet is, everybody agrees not yet is right, because there were all kinds of issues here. Certainly people aren't ready to do that. And the variations were a problem. People who have to leave, is that where you guys are getting ready to go? Feel free. Okay, now, let's just finish up. So, when effective and proven safe, I would be in favor of using gene editing in germline cells to avoid serious genetic diseases. So if you take people who are carriers or whatever for cystic fibrosis or muscular dystrophy or Huntington's disease, would you be in favor, how many people would be in favor of, so you have these choices. You agree, yes, I would be in favor of trying to fix those using germline editing, right? Not treating the tissues, but treating the germline. How many people disagree with that statement and how many people, any comments? I don't know if people want to say, I don't want to, the nice thing about the PRS clickers was you could express your opinion without identifying yourself. You can't quite do that here. So, yeah.
So are you saying that would be like the changing the genes that may occur, like diseases may occur because of them, or like ones that you know are gonna get diseases?
This is a good question. So if, for example, somebody is a cystic fibrosis carrier, you have one bad gene and one good gene. Half of your kids are going to get the bad gene, and they're not going to get cystic fibrosis unless your partner also is going to contribute a cystic fibrosis gene. So if you were checking each of you and found out, oh, okay, we each have one bad gene, there's a 1/4 chance that my kids will have it, then you could be changing it and you would be preventing 1/4 of those from doing it. For diseases like that, it's also true that you can, there's other strategies, right? You can do pre-implantation diagnosis and only implant, fertilized embryos that will be okay. But for some kinds of things, there are situations where all the kids will get it. There's no chance to do it. And so let's say that we were talking about diseases like that. I think it's an extremely good question because these are important differences. Gene editing germline gene editing is considered so serious, and it's kind of like we should solve these problems in any other way before we consider doing this. So, okay, just to throw a couple other things into this. You could also consider, once you learn about the infective mechanisms, for example, what goes on in cells for HIV to reproduce. Or, this is not known, but Ebola, which has, roughly, depending on the strain, maybe a 90% mortality if you're infected. We don't know enough yet, but once you find out how that works, you could potentially be making changes in the human germline that would make you resistant to Ebola infection. Okay, so another kind of possibility would be to consider doing such changes. Now, here's a little more subtle thing for you to think about, but this kind of thing will get talked about. So I don't know, are people aware of this the apolipoprotein E gene and being a risk factor for Alzheimer's? You read about this some. So the notion of this is you're like, let's say in this case we're referring to 3 different possible variants of the apolipoprotein E gene that you can have. And you're function pretty normally with any one of these 3, and they're present at reasonable amounts in the population. But people have found that if you have, if you're heterozygous, if you have one copy of the ApoE4, that particular variant, and something else, then you're 3 times more likely to get Alzheimer's than people who have this variant, ApoE3 only. And if you're homozygous, if you have 2 copies of ApoE4, you're 15 times more likely to get Alzheimer's. Okay? So one kind of idea is, all right, and in fact, ApoE, this other one may even be protected, may make it better than what most people have, the ApoE3. So the question there would be, okay, should you edit germline cells and convert all the ApoE4 alleles to ApoE3 so that you're reducing everybody's chance of getting Alzheimer's? Comments? So the good part, I mean, the good news, I think, is evident, right? It's like, well, sure, if I could wave a wand and say everybody is going to have, you know, a much lower chance of getting Alzheimer's with no other consequences, great. But I think the mystery in all these things is, well, but are there other consequences, right? Well, how will we know, and what could they be, and so forth? Do you guys know there's a famous gene where having it can make you sick if you have 2 copies, but sickle cell, right? So this is a kind of a classic example of this kind of thing, where the sickle cell gene, if you have 2 copies, then you have these sickle cell funny-shaped blood cells, and you have pretty serious disease and can die. And so it's kind of like, why is it so prevalent in the population? Why has it not been eliminated? And the answer as to why it's not been eliminated largely is because if you're heterozygous, you just have 1 copy of the sickle cell gene and 1 normal copy, you're resistant to malaria. And so this has been huge. There's like 580,000 people a year die of malaria. So in Africa, having 1 copy of the sickle cell gene is a tremendous blessing. You know, so if you were to eliminate that thing, thinking, oh, I want to get rid of sickle cell disease, then all of a sudden everybody dies from malaria. You know? So there's issues in all these kinds of equations about what you should do. When you get into really what people would call a slippery slope, is like, okay, what if after, 50 years, 100 years, 300 years from now, we learn about how to control muscle mass and height and intelligence and personality, then are you going to be in favor of messing with germline genes or not? And how do you write rules that would govern what you think is a good idea and what you think is a bad idea? Who's going to be the authorities that oversee the whole process? Will the authorities be able to oversee it everywhere, or will there be rogue laboratories that will give you things if you go and pay them and so forth? So this is the kind of controversy that's just blossoming, and which I think is going to be just huge for the next few decades. So I brought along 2 very short papers. One is 1 page and one is 2 pages. You can just have them, and I highlighted some parts of it. One of them is a guy who I would say is a fairly sober person who says, here are some of the advantages, here are some serious questions. The other guy is saying, don't cut it off. We need to have research, we need to be open, we need to encourage this. So one person is really kind of put on the gas pedal, the other guy is, let's play with the brake and the gas pedal. And in very brief summaries, then we'll give you some of the things to kind of think about. So if you want to take those, I'll leave those out here. That's it.
Thank you so much. If any of you have questions about your lab or need to talk about anything, I'm going to hang out in the hallway for a few minutes because I know there's a class in here. But pass your trash out, thank our presenter on your way out, and our next one's February 23rd.
Oh yeah, thanks. Please turn in the papers and I'll be